Welcome back, everyone. I'm Jonathan Weitzman. I'm just going to give my 3-second introduction. I'm going to do a shameless plug. So if anyone has not seen the Lonely Pipette podcast, we just launched episode number 4 today. Fiona Watt from the Medical Research Council and King's College in London. If you haven't, then please subscribe and please listen to the Lonely Pipette podcast and send me your feedback. And I'm really excited to have Nikos with us today. So I haven't heard Nikos speak since his job talk. So he gave a talk for his job, I guess it was about 1.5 years ago. And since then, so the committee was really, really impressed from the beginning. Since then, he's gone on to win an ERC grant. And we're really excited that he's going to be here really, really soon to start his lab at G.E.N.E. And the whole point of this seminar series was to really offer a platform for research on campus to be more visible. So he's not yet research on campus, but he will be research on campus very, very soon. So thanks for coming, Nikos. And this is a great way to think about something other than the American elections. So Letitia is going to introduce and give you all the rules. And thanks everyone for coming. Please come back. The next one is going to be in a month's time. And please come back, tell your friends, and send feedback. Thanks.
So hello everyone, and so thank you for being here. So as Jonathan said, I'll start by making a quick recap of the rules. So first, your camera and mic are off, and please do not turn them back on. So our speaker will share his screen, and so I advise you to use the speaker view on the top right of your screen to fully enjoy the seminar. At some point of the presentation, you can ask your question in the public chat, and I will read them out loud for the speaker. So I was supposed to introduce our speaker, but Jonathan did it perfectly. So we're very happy to welcome Dr. Konstantinides, and is going to present his research on development and evolution of neuronal diversity in the Drosophila optic lobes. And so I will let him start.
Okay, thank you. Thank you very much for inviting me to present. I also want to meet you guys and get slowly introduced into the research in Paris. As Jonathan said, I will be joining the G.E.N.E very soon, COVID allowing. And I would like to present to you today a little bit of mostly what I've been doing during my postdoc in Claude Desplan's lab at NYU, but also a little bit of an outlook of what I plan to be doing in the future in my lab. As I was telling Leticia earlier, I have split my presentation in smaller parts because I find it very hard to focus on Zoom for a long time. So I think it will be easier if we keep them in smaller parts. And then if you have any questions in between, please write them in the chat, as Leticia said, and we can also have some discussion during the presentation. So my interests are in neuronal diversity. So in neuronal diversity, and neurons are the most diverse cell types in the animal body. And of course, this difference in morphology has been appreciated for many years. And here you can see many different examples. But of course, we know that they're very different in terms of function, physiology. And lately, we've been learning more about their differences in molecular identity. And what I'm really interested in understanding is how this neural diversity develops and how over the few weeks or months of embryonic development, but also how it has evolved over time. And the reasons behind these interests, there are many. First of all, my specific interest is that it's very cool. It's actually pretty impressive to try to understand how such a complicated system develops and also how it has evolved. And here, for example, you can see the Drosophila brain, which you will see a little bit later as well. But also, as you imagine, it's much more complex if we look into vertebrates. Moreover, it's also a timely subject because lately more and more research has been focusing on developing in vitro neurons and glia that could be used for in a potential for neuronal therapy. And of course, it makes sense that only if we understand how this happens during real development, during the, in vivo, we can only recapitulate it in vitro. Finally, I think it's also very important for neuroscience because I believe it's necessary to understand brain function. And historically, the brain has been parallel and has been tried to be explained, given the most contemporary machine and the most advanced machine, whether this was a clock or hydraulic systems, the telegraph, the computer, and currently artificial intelligence. But there is no reason to believe that a system that has been evolving functions the same way as man-made things like the ones here. So I think that for many different reasons, understanding this, the development and evolution of nervous systems is interesting. And the system I've been using to track this question is the Drosophila optic lobe. So this is the visual part of the Drosophila brain. And it's located in the head of the fly directly underneath the eyes. And if you do a cross-section of the head, you can see the eye, which is here the photoreceptors, and this is the visual part of the brain, the optic lobe, which you can identify the different ganglia, which is where the neurons meet and form synapses, and they are surrounded by the neuronal cell bodies. And the reason why I think the optic lobe is there are many reasons why I think the optic lobe is a good system to study neuronal diversity. The first one being that neuronal diversity exists in the optic lobe. So research from over 30 years ago has shown that there are at least 100 different morphologically identifiable cell types in the brain. So here again, you see the different ganglia here, and you can see the different neurons that project to the ganglia and then project, forming different neurites and projecting into different levels of the ganglia and finding different synaptic partners. And here you can see different examples of these neurons. And these are only a handful, and we know that there are more than 150 different neuronal types. The other advantage of the optic lobe is that we have a pretty good understanding of its development. And we know that in order to generate all these different cell types, it's three mechanisms that come into play. And one is called temporal patterning, spatial patterning, and a notch binary cell-fate decision. And I will go into more detail in them. What I would like to highlight here is that this is by no means a peculiarity of the Drosophila or of the optic lobe in particular, but these mechanisms, although they were initially identified in flies, have been found to be at play also in vertebrate systems like mice and humans. So this, I want to tell you what I mean by temporal patterning. So this is a neural stem cell. So the neural stem cells in flies are called neuroblasts. And the neural stem cell, when it's born, it expresses a gene. In that case, we know the gene is called homothorax. And this neural stem cell divides to self-renew and generate a ganglion mother cell, which is an intermediate precursor, which gives rise to two neurons. And these two neurons are different from one another based on the presence or absence of notch activity. As the neural stem cell ages, it expresses a different transcription factor, which is called ILS. The neural stem cell divides again to self-renew and generate two different neurons, again that are different from the first ones because of the expression of this transcription factor. And this process goes on and on until the neuroblast terminally differentiates and stops dividing. And what is important here is that here we only have one neural stem cell that, by using this method of temporal patterning, so this temporal expression of transcription factors, as well as this notch binary cell-fate decision, is able to generate at least 10 different cell types. And the second mechanism that I told you is spatial patterning. This is something you're probably more familiar with and probably you've heard. So for example, the spatial patterning of the anteroposterior axis from the Hox genes. Similarly to this spatial patterning, this is the horseshoe-shaped epithelium where the neurons come from. And it's divided into domains, which are color-coded here. And depending on which domain they come from, they assume a different identity. So these are obviously two completely independent mechanisms and completely orthogonal. So if you put them together, you can immediately see that we can generate at least 30 different cell types using these two different mechanisms. And as I told you, it's important to note that this is not, these are examples that I'm showing you from the Drosophila optic lobe, but this is very much generalizable to vertebrates as well. So this is actually, and I want to actually tell you a couple of definitions so we're all on the same page. So this is what is called the specification of the neuron during neuronal development. So you can imagine, if you imagine that this is a neuron, this is the immature neuron that is specified by the temporal and spatial patterning, which is information that comes from its ancestors. Then the neuron differentiates to assume its final form and function in terms of morphology, but also molecular identity, i.e. to express the genes that are important for its function. And then these neurons has to maintain the expression of these genes and its morphology in a process that is called maintenance. And this also is something, as I will come back again, is something that is also shared between obviously vertebrates and invertebrates. So this will be more or less the outline of my talk. So initially I will mostly talk to you about development, which is what I've been doing in my PhD. And I will tell you 3 short stories about each of these different steps. And then at the end of my presentation, I will tell you about my interest in how to understand how these mechanisms have evolved. So the first one will be about the specification of neurons, and I already presented this to you. So this is the temporal series of transcription factor expression in the neural stem cells. And what I didn't tell you is that these genes that are expressed were identified by transcription factor screens using antibodies. So this means that labs, including Claude's lab, have screened banks of transcription antibodies against transcription factors, and they came up with these 5 transcription factors that are expressed in a temporal manner. However, these banks are not exhaustive, and we don't really know if this is indeed the whole story. Moreover, we know that there are more than 30 neuronal types that are generated from this in this structure. So we expected that there are more transcription factors expressed during this temporal series, and that's what we wanted to address. And for this, we use the technique which you might have heard about, which is called single cell sequencing. So I would like to introduce it to you because I'll be using it in my entire presentation. And this is a technique essentially that allows you to get the transcriptome of a single cell. And it works usually it uses microfluidic devices that are able to isolate beads like these big balls that you see here with cells, which are the ones that you see coming from this pipe here, into an aqueous droplet in an oil background. The cell is then lysed in this droplet, and the RNA is hybridized on the beads that carry specific barcodes that can allow us then to identify the specific cell that the transcript came from. This is a very powerful technique that I'm sure you have heard about and allows you to get single cell resolution in the transcriptome. And that's what I've been doing, I've been using to try to address this question. And I will show you how I've been using this. Actually, I've been taking advantage of a unique characteristic of the developing Drosophila brain. And here you see a cross-section of the developing optic lobe in the larval stages of Drosophila. So initially, in mid-third larval stage, the surface is populated by neuroepithelium, which is marked by NE here. And when neurogenesis starts, the neuroepithelium is transformed into neuroblasts in a process from medial to lateral. So this is the first neuroblast to be born, the second, the third, and so on and so forth. And because of this process, at a specific time point that you see here, which is in the late stages of the third instar larva, you have neuroblasts of different ages. So essentially, you have different neuroblasts of different ages that express the different transcription factors that I showed you here. So what we thought of doing was to try to isolate the cells from this stage, perform single cell sequencing, and try to identify the transcription factors that are expressed. So that's what we did. We did the single cell sequencing. We identified based on known markers the cells that are the neural stem cells. And then we wanted to use something that's called the trajectory analysis to try to order them. Obviously, when you dissociate the tissue, you lose this structure, but we wanted to use bioinformatic analysis, which is called trajectory analysis, to order them in a temporal manner. And I will talk to you more about that in a couple slides, but essentially this would allow us to identify all the genes that are expressed temporally in this axis, and I then select the temporally expressed transcription factors. So I'll come back to that in a couple slides. So I will be showing you also these plots quite a lot during my presentation. So this is a two-dimensional representation of single cell sequencing data. You might have heard of two techniques that people are using. They're called t-SNE and UMAP plots. These are just two different dimensionality reduction techniques. I will be showing you UMAP plots today. And essentially in these two-dimensional plots, every dot is a single cell, and they're organized in these two dimensions based on their transcriptome, based on the expression of different genes. So the assumption is that genes that are closer to one another, cells that are closer to one another, have more similar transcriptomes. On top of that, on this plot, you can project the expression of specific genes using different colors. So for example, here I'm showing you a gene which is called DEDPAN, which is expressed at differing levels, 0 wherever you see gray, and higher expression wherever you see red. So we can use these methods to try to annotate all these cells and see where they come from. So DEDPAN is a marker for neural stem cells. So we know that these are the neural stem cells. Then we have a marker for these intermediate precursors that I told you earlier, which is called ASENS, which is expressed in the neural stem cells and in these intermediate precursors. And finally, you have LAV, which as a protein is only expressed in neurons, or mostly expressed in neurons, but you can see that the transcript also persists, can be identified earlier. So what this shows us is more or less that you have an axis of differentiation from right to left, here having the neural stem cells, the GMCs, and the neurons. So as I told you earlier, what we decided to do is identify the neural stem cells based on the expression of DEDPAN and use something that's called trajectory analysis. And trajectory analysis, the way it works is that it assumes that as a neuron develops or as it differentiates, it expresses, it has incremental changes, small incremental changes in the transcriptome, that if we manage to capture by the single cell sequencing, we would be able to order these cells in a trajectory that would recapitulate their differentiation. So for example, this would be what you expect to get. So this is a trajectory of cells. Again, these cells, each dot is a single cell, and they're ordered by, as I told you, these small incremental changes in the transcriptome that this program, which is called Monocle, can identify. So if this was working as advertised, you would and this is the neural stem cells that I told you earlier, you would expect to see the genes that we know are expressed temporally in this series to be expressed temporally in this case as well. And here you see a heat map of these genes. Red and yellow is expression, blue and green is lack of expression. And indeed, you can see genes here, homothorax being expressed in this side of the trajectory, eyeless being expressed later, sloppy pair, dichetan, tailless. So indeed, by using this method, that by leveraging these small differences in the transcriptome, we can put them in an order that recapitulates completely what we know in development. So this is a very good positive control for the function of these algorithms. And then, of course, what I wanted to do, as I told you, is to try to identify transcription factors that were unknown and might be expressed temporally. And this is, I screened all 700-800 transcription factors or Drosophila. And these are the 12 or 13 transcription factors that are found to be expressed temporally. So with green, you can see the already known ones that were blindly found again, obviously. And these are the ones that were expressed, that were found to be expressed temporally, that were unknown. And of course, the next step would be to verify that these transcription factors are expressed temporally in the brain. So here I'm just reminding you, this is the developing optic lobe. Here you can see in the surface, as I told you, these are the youngest neural stem cells. These are the older neural stem cells. So by doing stainings between known and unknown transcription factors, we can know where these newly identified transcription factors are expressed. So the first example I'm going to show you is homeobrain. So homeobrain is a gene that was identified bioinformatically. Sloppy-Part was already known by a previous study. And you can see here, if you look at the surface, you can see indeed that homeobrain and the neuroblast stage goes this way. So we're looking, we're looking this structure from the top. So homeobrain is expressed before Sloppy-Part, as you can see here. So homeobrain is in green, Sloppy-Part is in purple. Another example here is eyeless and scarecrow. So eyeless is the blue neuroblast here, and scarecrow was identified to be expressed after scarecrow. Scarecrow was found to be expressed after eyeless bioinformatically, and indeed you can see that scarecrow is expressed in neuroblast of older age. Using these stainings and for transcription factors for which we could generate or find antibodies, we were able to verify all the different transcription factors to be expressed temporally. So here you can see examples. So with purple here, you can see the ones that were already known that I told you in the introduction. And with green, you can see the expression of the the verified candidates. So here, this was the temporal series that was published in 2013. What I don't have time to tell you now, but I'm happy to discuss later, is that the authors in 2013, which were from Claude's lab, had found that the genes that are expressed in the temporal series, they're involved in gene regulatory networks that activate the next transcription factor of the series. So ILS activates Sloppy-Part, and they inhibit the previous member of the series. So Sloppy-Part inhibits ILS, and this way allowing for the series to progress. We also did the same analysis with our new transcription factors, and we found an enhanced temporal series that now we believe is also it's complete, because we had a way of comprehensively addressing the expression of all different transcription factors. And so this is the first part that of my presentation. And so we have identified an expanded temporal series of transcription factors, which we're now testing for its function. So we're trying to understand what kind of neurons come from each of these temporal windows. What I think is very important, and it's very also important for the future of my research, is that we can use such techniques to identify all the temporal factors in the developing brain of any animal. And essentially, we can expand this approach to other species. The advantage of Drosophila, obviously, is that we have availability of antibodies and of genetic tools. But based on what I presented you now, even in the absence of such tools, we would be able to identify these temporal factors, even if we didn't know exactly the genetic interactions between them. And this is the first part. So if I don't know if there are any questions that you would like to address, even either in terms of the technique or the biology. Okay. So the second part of my presentation will have to do with differentiation of a neuron. So a neuron, as I told you, once it's born, it has already assumed its identity, so it knows which kind of neural type it's going to become, but then it has to express the specific gene, the appropriate genes, and acquire the appropriate morphology. So I will take you back again to this schematic of the developing optic lobe. So what I described to you is the neural stem cells and how you have neural stem cells of different ages. But in fact, this difference in ages is also reflected in the neurons. So as the neuroblast is aging, as I told you, they generate neurons. And here neurons of the same cell type are color-coded in the same way. So these are the red neurons that came from this neuroblast when it was alive and it was red. These are the red neurons that came from this neuroblast when it was red, and so on. So these were the first ones to be born, and these were the last ones to be born. So essentially, you have the same difference in ages that you see in the neuroblast, you can see them in the neurons. And this means having neurons of different ages and hence different level of differentiation. We can use similar techniques like I showed you earlier to find all the genes that are expressed in a temporal manner in the different neurons. And as an example first, so here this is, and also what I want to tell you is that this is the same plot that I showed you earlier. So this is the single cell sequencing from the larval optic lobe. I was lucky that at the time that I was doing this research, there was another postdoc in the lab, Neshek, who did the similar approach in a later stage of development. And this is the early pupal stages. And in fact, what you see is something that you might expect, given that I told you that the differentiation axis is this way, that all the neurons that are more differentiated can be found at the tips of all these branches here. So what I decided to do is I identify a neuronal type that we know well and we have studied in the lab, and try to see if I can study the trajectory of differentiation. And essentially, I did the same approach that I told you earlier. So I used Monocle to generate the trajectory. This is the trajectory that was known to, that was generated by the algorithm. And in that case, we didn't have good markers like the ones I showed you earlier for the neuroblast. So I used markers of the intermediate neuronal precursor and of the differentiated neurons to order this trajectory. So this is a marker, as I showed you earlier, for the intermediate precursors, and this is expressed in the left side of the trajectory. And this is a marker of the differentiated neuron, which is expressed at the right side of the trajectory. So I could order this way. And then I asked what kind of genes are expressed along this trajectory. And in fact, there are many genes that are expressed differentially. Here you can see it's a list of about 1,000 genes that are expressed at different stages along this trajectory. Again, to remind you, young neurons are from this side, all more differentiated neurons are from this side. Red and yellow is expression, blue and green is lack of expression. So you can see that there are many genes that are expressed at different stages of this trajectory. So what I did is that I grouped them, and then I performed the gene ontology enrichment analysis, and I came up with a process of differentiation. So the first genes that were enriched are genes that are involved in cell division and cell cycle, and essentially these genes, these cells here in the very beginning of the trajectory, are remnants of the intermediate neuronal precursors. Then you have a boost in translationally related genes, and these are genes that are involved in regenerating the translational machinery and the ribosome after the division. Then you have an enrichment in action guidance, which is and dendrite development, essentially when the neurons are trying to find their targets. And then the expression of genes that are more functional, that are related in neurotransmitter synthesis, synaptogenesis, cell adhesion, and even response to stimuli. What I think is pretty, this is absolutely, I mean, I think it makes absolute sense. Of course, a neuron has to go through this process in order to be able to function. What I found really surprising is that essentially these genes, these proteins, are not going to be used for at least 2 to 4 days, which is when the fly will either form synapses in mid-pupa or respond to stimuli in the adult, which is, I found what's pretty interesting that they're already expressed from the larval brain. Of course, what I told you, this is true from one specific neuronal type. So I wanted to see if I was able to generalize. So essentially what I did is that I went back to this, to the plot that I showed you earlier, and I plotted the average expression of all the genes that are involved in these processes. So here you can see the expression of cell cycle related genes, which as expected is enriched in the neural stem cells and the GMCs. Then you have genes that are involved in cytoplasmic translation. Obviously they're expressed everywhere, but they're mostly enriched in the beginning of the different branches. Then you have genes that are involved in action extension, and they're expressed midway through all these different branches. And just to remind you, these are branches that give rise to different neuronal types. And you can see that this is not only true for the specific neuronal type I talked to you earlier, but for all of them. And then genes involved in chemical synaptic transmission, which are expressed by the end of this trajectory. So essentially what we came up with here is a common path for neurons upon specification, where initially you have an enrichment in cell cycle related genes and cell division related genes, and ribosome assembly and translation related genes. Then you have an expression of genes that are involved in action guidance or dendrite development. And finally, by the end, you have genes that are involved in synaptic transmission, as well as regulation of membrane potential and other functional genes. So what is the take-home message from here is that neuronal identity is largely implemented within the first hours of neuronal life. As I told you, these genes are expressed much earlier than I would have expected, at least. And all neurons follow a common path of differentiation upon specification. And this, again, I want to highlight, is not a peculiarity of Drosophila. In fact, we believe that this is a common feature of neurogenesis in animals. And this comes from actually a study that was published last year by the lab of Denis Jabodon in Geneva, where essentially what they showed here, APs are the neural stem cells. These are the apical progenitors of the mouse cortex. And what they show is that this temporal progression of neural stem cells, equivalent to the temporal series that I showed you earlier, leads to the generation of specific neurons that can acquire each of these roots. But then when this neuron acquires a specific root, it follows a precast path of differentiation. And I want to borrow some of his words from one of Denis Jabodon's work from his presentations. The way he describes it is that the neural stem cells decide what kind of cake you're going to make, whether this will be a lemon cake, an orange cake, or whatever. And this decides what kind of ingredients you're going to use. But once you decide what kind of ingredients you're going to use, the process of making the cake is the same, even using different ingredients. And this is what we also see in Drosophila. And I believe that this is a common for all animals. Actually, in fact, we were lucky to be able to get our hands on some data from human cortical neurons of fetal week 19. This is close to the end of neurogenesis in humans. And in fact, what we saw is that the process is remarkably similar with few differences, but initially you have the expression of cell cycle related genes. Then you have a boost in genes that are involved in axon guidance and dendrite development, and then genes that are involved in chemical synaptic transmission and regulation of membrane potential, which are the same terms that I showed you earlier from Drosophila. And this is, in fact, work done by a master student from Paris that worked with me during the summer. And this brings me to the third part of my presentation, which is involved in the maintenance. And this is actually, chronologically, this is the first part I did during my PhD. And probably this is what Jonathan heard about during my job talk at the Jacques Monod. Essentially, what we wanted to see here was to try to understand how different transcription factors are involved in the expression of specific genes in differentiated neurons. And in order to do that, we used, again, as I told you, there are about 100 to 150 cell types in the adult optic lobe. So we wanted to get access to the expression of all these transcription factors in the adult neuronal types. So for this purpose, we used, again, single cell sequencing from the adult optic lobes. So this is now a Disney plot. This comes from the adult brain. So immediately you can see that the clusters are much better resolved than what I showed you earlier. There is no continuity between most of these clusters. And the reason is because these cells are already differentiated and they differ from one another. So essentially what we did is that we generated an atlas of the expression of different cell types in the adults. And this gave us access to the expression of specific transcription factors in each of these cell types. So then what I wanted to do, as I told you, is to see how different transcription factors may be involved in the expression of specific neuronal characters. So essentially, I used machine learning modeling to be able to generate a model that would take transcription factor expression as an input and output the expression of the entire transcription of a specific cell type. And the way I did it, so this was done with an earlier version of the dataset where we only had 52 clusters. Now we have almost full resolution. And essentially what I did is that I split my dataset into two sets. So one of them was a training set, which is composed of 39 clusters. and a test set composed of 13 clusters. And then I trained a machine learning model, as I told you, to get as an input transcription factors and output the entire transcriptome of these cells. And then I used this model to try to predict the transcriptome of the clusters of the test set based on their expression of transcription factors. What is very important to note here is that the model itself has not seen the transcriptome of these cells. So it has only seen the transcriptome of these cells, and that's the idea of having these two sets. And this is how the model performs. So on the y-axis, you can see predicted gene expression. On the x-axis, you can see actual gene expression. And for the neuronal cluster, the model performs remarkably well in predicting expression of specific of all the genes. It performs less well in the glia, and we believe that the reason for that is because we have fewer glial clusters. The model was improperly trained. So, but the main question that I wanted to address is, if I have a specific gene of interest, can I predict the transcription factor that may be regulating it? So essentially, this would be the opposite question. And in order to address this question, I focused on neurotransmitter identities in the optic lobe. So here you can see the different neurotransmitter identities. So we have cholinergic, glutamatergic, GABAergic, and dopaminergic cells in the optic lobe. I identified genes that are expressed exclusively in these cell types. So this is the cholineacetyltransferase, the vesicular glutamate transporter, GAT1, which is an enzyme necessary to make GABA, and the vesicular monoaminergic transporter. And using the model that I described to you earlier, I predicted the transcription factors that essentially are best correlated with these genes. And these are the transcription factors, the top key transcription factors. Their names are not so important, but essentially what we wanted to do later is to try to understand and to try to confirm that they're indeed regulating these genes. And in order for that, I used the genetic toolkit of Drosophila. So here you can see the optic lobe. So these are the different ganglia, and you can see the expression of cholineacetyltransferase in a wild type brain. You can see it's pretty broadly expressed. So upon downregulation of Apterus with an RNAi, we saw a huge downregulation in the expression of CHAT, which showed essentially that Apterus indeed is regulating directly or indirectly the expression of CHAT. I did the same for the other genes as well. So here I present to you just the results of a real-time PCR. And we found that indeed, when we downregulate the predicted transcription factor, using RNAi, the expression of this gene is also reduced, which showed indeed that these transcription factors that we identified regulate the neurotransmitter phenotype of the different genes. The other thing, and we're now working hard to also expand beyond these specific genes. The other thing that was also actually pretty interesting is that you might have noticed that although in some parts of the ganglion, you can see almost complete elimination of expression of CHAT. In other parts, it seems like it's unaffected. So what we actually observed is that indeed in some cell types, CHAT is regulated by Apterus directly or indirectly, whereas in others, the expression of CHAT is regulated by a different transcription factor. So we showed by correlation analysis, we showed that this is not only true for neurotransmitter genes, but also for most of the genes that are expressed in neurons. And we call this phenotypic convergence in that the cells, converge to the same expression using different transcription factors. And we believe that this is also using different developmental pathways. So the take-home message from this part is that we use single-cell sequencing to describe the neuronal diversity in the fly optic lobes in the molecular level. And as I told you now, we have almost full resolution of the different cell types. And we were able to use this data to identify transcription factors that regulate the expression of specific genes, and in particular, in our case, that regulate neurotransmitter identity. And finally, what I told you in the last slide is that we uncovered an extensive degree of phenotypic convergence, which has since been observed in other neuronal systems, including mice. And this brings me to the last part of my talk, which would have to do with evolution. And the main question that I would like to understand, the first question actually that I would like to understand, is how fast neuronal types evolve. And we actually have no idea how fast this happens, and we don't know in any neuronal system, in any group of animals, how different the neuronal type composition is in between the similar structures. And that's what I would like to do. And I will focus on insects that are related, that are close to Drosophila, because we have a very, very good understanding of what's happening in Drosophila. So I will show you an example of how I plan to do this. These are some preliminary data that I got from the housefly, Musca domestica, and how I compared this to Drosophila. One of the main problems of aligning transcriptomic data is something that has to do with the fact that come from different species. So this is a toy example of getting transcriptomic data from different species. So one is exemplified here by a circle, and the other one is by a triangle. And by color, you can see what would be homologous cell types. So if you perform single cell sequencing, what you expect to see is you expect to see clusters of cells that are different based on their cell type, of course, but also based on their species of origin. And this makes sense because obviously these species have diversified millions of years ago. So the goal would be to use specific algorithms to align these data sets and essentially eliminate the species-specific differences. And for this purpose, what I used and I'm planning to use is something that's called batch effect reduction algorithms. So these batch effect reduction algorithms are used to eliminate batch effects that come from different batches. This means that that come from different experiments, from different researchers in different countries and so on. But we can also imagine that the two species, the fact that they were evolving separately, we could imagine that they represent two different batches and use such algorithms to try to eliminate the species-specific differences. And this is, as I told you, a toy example. This was before we actually did the experiments, but in fact, they look fairly similar. So this is this data, the pink ones that come from Drosophila, and this is the data set that I've already showed you. And this data come from Musca. This is a smaller data set that we generated. And indeed, when you try to merge these two data sets, the first axis of differentiation is species. However, once you use one of these algorithms, you can essentially eliminate these specific differences. And you can see that now the two data sets overlay pretty nicely. And this is not completely random as could have been. But in fact, if you look for the expression of specific genes, so here you can see the expression of VGLUT, which marks glutamatergic genes, and GAD1, which marks GABAergic cells. You can see that indeed both Drosophila and Musca cells cluster together. And here you can see both Drosophila and Musca GABAergic cells, both Drosophila and Musca glutamatergic cells. These are the cholinergic cells, and these are glia. And the goal of that is to then go into the very specific, to the different neuronal clusters and identify neuronal cells that are represented in both species, like the one you see here, or ones that are enriched in one species or the other. And then try to understand what kind of differences exist between these neural types, and importantly, how these differences arise during development. My second interest is to try to understand how developmental mechanisms also have evolved. And the main example I'm planning to use is temporal patterning. So I showed you earlier how you can use single cell sequencing and trajectory analysis to rediscover, essentially to discover the temporal series, or the genes that are the transcription factors that are expressed in a temporal manner. So the goal would be to look into other insects and try to identify differences. So one difference that one could identify is the addition or the elimination of a temporal window, which would have a consequence in neuronal diversity. Or you could imagine that you could have a duplication of part or the entire lineage. So my goal is to go into different insects and try to understand how this temporal series has evolved, as this is something that we really have no idea how it happens. And these are my two main goals that I will be working on in my lab. And with this, I would like to thank Actually, all the people in Claude's lab. So this is Claude. This is our lab. This is my funding. So this is my old thank you slide. My new thank you slide is a bit more minimal. I guess I don't need to show you where the Jacques-Monod is. You probably need to show me at some point around. And this is will be funded by the CNRS and the ERC. And I'm happy to take any questions.
Tu veux faire les questions?
Thank you very much. So there are questions from Jonathan. The first one is telling that you only look at gene expression, and do you think that the 3 steps, specification, differentiation, and maintenance, also involve post-transcriptional and post-translational changes, and how could this be integrated with your data?
Yeah, so yes, so absolutely, of course it is. And there are many different aspects. So first, the one thing that I can say in terms of differences of transcriptome and proteome, there are 2. So first of all, we know I have tested the expression of some of the genes that the transcript is present in very young neurons, and I do not see the protein. So these are the genes that I mentioned to you earlier, which is cholinesterase transferase and the VGLUT. So both of them, although the transcript is there very early, even from the larval stages, we do not see the protein. So we know that there is some kind of post-transcriptional regulation. The second example that, as you might have noticed, I grazed over very quickly and didn't even mention it afterwards, is spatial patterning. And in fact, so let me go back. So if you remember, spatial patterning is the second, oops. So spatial patterning is the second mechanism that is involved in the generation of neuronal diversity. And in fact, when we look at the expression of these genes in neuroblasts and in neurons, we don't really see them being expressed. And in fact, work from other labs and in different systems in the ventral nerve cord has shown that they probably act in the level of the neuroepithelium. They affect the chromatin, they alter the chromatin, and then they stop being expressed. So this is something we don't know. I am not planning for the time being to work on that. There is a PhD student in Claude's lab that is working on this question to try to see how these effects are being translated into neuroblasts. And how this can be done, it depends on the level of detail. I guess in a level of detail as broad as this, one could and actually that's what they're planning to do, is to combine a TAC-seq with RNA-seq and try to see if they can resolve these differences. Yes, so I don't know if this covered the question.
So there's another question from Yvon. He asked if you could re-explain how you train the computer to identify new transcription factors.
Yeah. Wait, so is this about the trajectories, or is it about the trajectory or about the machine learning?
Machine learning.
Okay, so essentially, I'll try to find it. Yes. So essentially, so the machine learning model I used is called random forest. But we could imagine it's something like a linear model, let's say. And the way it works, it takes, for each specific gene, it tries to generate a list of coefficients for each of these transcription factors. And whether the coefficients are positive or negative is whether they activate it or repress it. And if the coefficient is large, it means that the effect is large. So essentially, the output of this model is for each gene, you get a list of transcription factors that have a high effect. And this effect is essentially a metric of the correlation of expression in the training set. So essentially, you try to generate such equations between transcription factors and genes. And then you use the coefficients that are generated from the training set into the test set. So you give the coefficients to the transcription factors and try to predict the expression of each gene separately. So now it doesn't work exactly like a linear model. So you don't exactly have linear, you don't have coefficients per se, but it's a similar way that it works with random forest. And I'm happy to talk about later in more detail if needed. Does this answer the question, Ivan?
Okay, yes.
Okay.
So another question from Jonathan about the patterns of waves of expression. He asks if they are similar in other tissues and other differentiation programs, like making cakes in different tissues.
I guess he means outside of neuronal tissues. So this is something I don't really know. So in fact, this complete division of specification and differentiation, I'm not sure can be generalized in other tissues. So I would expect it. I would expect it to be different, but I think it's outside it's out my expertise. I don't really know about other tissues what would happen. I know that I really don't know actually.
Okay, so Jonathan is saying that every tissue makes cakes differently and there are more than one way to bake a cake.
Yes.
So there is a question from Emmanuel. Comparative transcriptomics in mammals showed that timings of expression are rather conserved for transcription factors and in general for protein coding genes. Do you intend to address how the flies' non-coding genomes, at least non-coding RNAs, could explain the evolution of neurons in flies?
Sorry. Oh, okay. Yeah, so this is an interesting question, and it's also something that I don't know exactly how to address. So I think we might have the technology to do that, but we don't have the knowledge. So something that is interesting is that what I showed you as a temporal transcription factor series in Drosophila, this is completely different with what happens in invertebrates. So almost none of the genes are used in vertebrates in a similar way. The only similarity that exists is gradients of RNA binding proteins. And I think this might show an importance of non-coding RNAs in this process. But also, most of the non-coding RNAs that we know from vertebrates are not necessarily conserved. So it might be hard to address, but I think it's interesting, and it's very possible that there are such effects of non-coding RNAs also in the specification of Drosophila neurons.
So the next question is from Aurélien. Could you please re-explain the plots about single-cell transcriptomic?
Yes. So let me. Yeah. So essentially, let's go to this plot, which is a little bit easier, but it is. I think it's as in. So okay. So as I told you, so what you as an output of a single-cell sequencing experiment, you get information for thousands of genes. So in Drosophila, it's like 13,000 in specific cells. So you could imagine that in order to represent this in the space of gene expression, you would need 13,000 dimensions, so one for each gene. So people have invested a lot of time in order to try to make a 2-dimensional or 3-dimensional representation that keeps as much information as possible. Obviously, this is not all the information because we cannot really reduce all these dimensions into 2. So essentially, they're using something that is called non-linear dimensionality reduction techniques, which I showed you 2 today. So one is t-SNE and UMAP, and these are the 2 that are mostly used to generate coefficients, 2 coefficients. So one is t-SNE 1 and one is t-SNE 2, that would allow us to present this data in a 2-dimensional space. So essentially, what you get here is you get genes. You get each of these dots. So you cannot see them, but all of them are dots. Is a single cell, and their transcriptome is somehow reduced into these 2 dimensions. And they try to keep as much information as possible. So in the end, indeed, you have clusters of cells that transcriptomically are similar. So they have similar gene expression patterns. and these are color coded here. So, and this is one technique is called TSNE, the other one is called UMAP, but essentially they have the same idea of projecting all this diversity in 2 dimensions. Laetitia, did I answer your question or Aurélien?
It was Aurélien. Okay, he's not saying anything, so I think it means yes.
Okay.
Oh, yes, he said yes, actually.
Okay.
So next question is from Tanya. Could you precise how you did the alignment of the different neural types composition and the comparison between Drosophila and Mosca?
Yeah, so I can briefly explain that, but in fact, so the math behind it is pretty complicated. So I can point to you the paper where it was published, but essentially what these algorithms generally try to do is they're trying to, so they assume that the tissues that you're comparing are similar enough. So they assume that they're the same essentially. So they're made to reduce batch effect, which means that you're studying the same tissue in different labs, let's say. So essentially, what they're trying to do is to try to identify common patterns. So they try to identify groups of genes that are expressed commonly and trying to generate a common space of expression and project the expression of these cells into this common space. So essentially, they choose a few thousand genes that are used to generate a common gene expression space, and then you try to put these into this common gene expression space. I'm not sure I can explain it very well, but I can definitely point you to the paper that does that. But it's pretty complicated.
Thank you. Another question from Leos Portes. I'm not telling it well, maybe. Is there a future in neuropathology?
I don't know how to answer this question. Yeah, I don't know what they mean by neuropathology. But if this means in neuronal therapy, I'm sure there is a big future. I think we have now a lot of tools. I mean, we're in the very beginning and we don't know much, but I think we're in a very good time to study different things and try to focus on neuronal therapy. And there are different approaches to do that. If this has to do with neurodevelopmental disorders, which is something that I'm more interested in, well, it's a bit more relevant. Also, I think there is a lot of future because now we're understanding a lot about development, and we're understanding in a more general aspect how different genes affect different developmental processes and how deregulation of these genes can affect specific disorders. But I don't know.
So I think we have one more question from Philippe. Mm-hmm. If the transcription factor identity of a neuron is established early on, how can different cellular processes be regulated during development, such as targeting, synaptic partner matching, etc., all of which happen at different developmental time points? Also, if post-transcription regulation is taking place, how is this regulated in time? Do you think input from the environment, the developing neuron encounters, can feed into the activation of such post-transcription regulation? And if yes, how?
Yeah, so absolutely. So we know that all these processes are also regulated in time. And of course, once making a cake, you have to add the correct ingredients at the same time. So obviously there is input from the environment that comes out. And I guess Philippe knows that egg Dyson is something that plays a role in regulating these processes. So we know that they're regulating in time by the environment. And whether the input from the environment could affect post-transcription regulation, I guess so. I wouldn't have a hard time imagining that this would be the case. but if we have a specific example for that, this is something I do not remember at the time. I don't think we have a specific example for that, but we know that environmental input affects the neuronal development.
Okay, so I think it was the last question, so we'll stop here, and we thank you very, very much for this seminar, and we are very happy to welcome you in Paris as soon as possible.
Thank you.